Yeah, I'm sorry about the last lecture not being recorded. That was mostly my fault because I forgot to stop it. So it ran literally all day and then it couldn't save it on the cloud because it was such a large file. So that did not work out. So hopefully I remember to stop it this time. Okay, maybe let's get started and people can come in as they, whenever they're ready. So I wanted to, so today is really the last lecture where we're going to be talking about translation and all the aspects of translation. And then the idea is that on Thursday, we talk about how we incorporate all of the different levels of gene regulation that we've discussed and ways in which you can study this on a larger scale, so more genome-wide. And then that will be the last lecture. So the final exam will cover lectures 12 through 16. There's no lecture 17 as it says in the syllabus, but that's also been updated initially. So I just kind of compressed several of the lectures on translation to be able to accommodate those lectures. The other thing is that I want to mention, and I'll also add an announcement, is that on Friday, at the start of office hours, which is at 3, the idea is to have a whole class review session because that's the only time that actually works for me. And I will run it for as long as people can. So it can go beyond 4 or 5 or whatever time. So I'll try to at least be there between 3 to 5. The idea is that is to go over a more compressed version of a study guide for lectures 1 through 11, instead of you guys studying every single thing that covered in all those lectures, because that's only going to account for about 50% of the final exam. And the remaining 50% will be on the lectures that you haven't been tested on yet, which is lectures 12 to 16. Okay. Any questions about that? Hi, yes, Shella.
So I understand that it's 50% new lectures and 50% the previous lectures, but since we do 3 parts of the exam, I'm a little kind of confused on how it's going to be broken up, like, because I know we don't have that many questions to begin with. Yeah,
so it's just incorporated within those 3 different sections.
Okay.
Yeah, I mean, it is because it's the final and you have more time. It's definitely going to be longer than exam 2, maybe a little bit longer than exam 1, but not significantly longer. Most people from previous years have been able to finish the final within 2 hours, but that's assuming that, you know, that was in person, right? And you can see that you're much faster if you're doing these exams in person than online. So I anticipate that everyone should be able to get all parts done within the 3 hours, but the questions are just kind of all aspects, all of the lectures in the class is just kind of integrated, but you will
see. Okay, so we should expect more integration problems to cover like all the previous material.
Yeah, I mean, you're going to see questions that are specific, but you will see for each section more questions on the last couple of lectures, right? So let's say, for example, you know, let's say we have 20 questions for the multiple choice, and this is just a random number, right? 10 of those questions will definitely be on the last set of lectures, right? And then the other 10 will be split between the other 2 lectures. You know what I mean?
Oh, okay. Yeah, that definitely makes sense. Okay. Thank you.
So, Any other questions? So yes, I'm not going to re-record the lectures. I think what I mentioned is please go over lecture 14. And if for some reason there is something that you're missing or you don't get, just let me know and send me an email. But yeah, they're definitely not going to be able to re-record any of the lectures that is missing, which is only lecture 14 as far as I'm aware. So yeah. Okay, so let's get started. For someone asked if there's going to be extra credit in the final. Yeah, so in the final, I usually throw in 2 or 3 different extra credit questions, usually 3, to kind of cover the 3 major areas of the class. It's it's an extra credit, it's optional. So if people feel like they want to answer it, they can answer it. It's not mandatory, and you'll see it towards the end of the exam. And they're usually very easy questions. So sometimes it's questions that you've seen before on previous exams that I found that many people might have a problem with it. So I just kind of put it back in there so people can kind of get it. So what those questions are right now, I have no idea yet. Yeah. So, okay, so let's get started. Do you have another question, Shayla, or your hands are, you still have your hands up before I start?
Oh, sorry, my hand was still up.
Okay, just making sure, yeah, thanks. Okay, so just a refresher on what we talked about in terms of the elongation cycle. And I mentioned that, there's all these components that are involved in translation elongation. But I think if you just split this into steps, right, I think it's going to be really easy to kind of put everything together. And this is for both bacteria and also in eukaryotes as well. And if you, again, summarizing this graph, what is needed, right? Thinking beforehand what is needed before the ribosome is bound to this mRNA, right? And so you know what have to happen before this can occur, right? And that's just thinking about that beforehand. And then during the elongation cycle, where you have all of these components that are coming in, adding the amino acyl tRNA with the amino acid attached to it, right? You're transferring the amino acid, and the cycle continues. And this will continue producing a polypeptide until the ribosome encounters a stop codon. So in prokaryotes, the stop codons are recognized by different proteins, and these proteins are referred to as release factors, or RFs. Bacteria have 3 of them, and they are specific depending on the type of stop codon. So RF1 recognizes the UAA and UAG, while RF2 will recognize UAA and UGA. Okay, so 1 and 2 recognize, both 1 and 2 will recognize UAA in some contexts. You also have another release factor, which is a GTP binding protein, or it has GTP binding capabilities. And this release factor is important because it really helps to facilitate the release of RF1 and RF2 once they've done their job of terminating translation. Eukaryotes, on the other hand, have only 2 release factors, ERF1 and ERF3, and again, recognize all 3 of the different termination codons. ERF3 is the one that has the similar GTPase activity as RF, the prokaryote release factor. Okay. And the way that these release factors work is that they really help to, because you would think that when you encounter a stop codon, there's no amino acid really for that stop codon, okay? So you cannot add an amino acid. So once a ribosome encounters the stop codon, you can see these are 2 experiments, and they're just graphically shown here in 2 cases, A and B, where you have a start codon with the start methionine attached to it, and you can add the stop codon, and also one of the release factors, in this case 1 or 2, which recognize the specific ones, and you can see that it's sufficient to release this FMET and terminate, and just release that one FMET, and that's it, right? So it's kind of terminating translation. And these were experiments that were actually done where they were adding these components to see whether or not you can actually release the amino acid that has been added with that ribosome. Okay, so that's how they facilitate termination. They really do so by positioning themselves in that A site that's empty, right? So the release factor will bind to those A site because again, you encounter a stop codon, there's no recognition for that tRNA to bring in amino acids. So instead what goes there is these release factors to do the job to destabilize the complex completely. And for the most part, when this happens initially, when translation, when the stop codon is encountered, is the ribosomes do not just spontaneously release the mRNA after termination. You need the help of additional proteins in order for this to happen. Okay, so in eukaryotes, the ribosomes are released by these EIFs, right, which is elongation factors, the initiation factors initially that you saw when we're doing translation initiation, and that's aided by several subunits. So all of these are this part of a subunit of this EIF3, and they help to really release the ribosomes from the mRNA. In prokaryotes, you have these ribosome recycling factors or RRFs, and also other types of elongation factors that are important for helping that ribosome to disengage from that mRNA. Okay, RRF is one of those that actually it looks like a tRNA, right? So it will bind in bacteria to that A site, and because again it's not a real tRNA, and even if it was a tRNA, you can't bring an amino acid because there's just stop codon. So it really sits there and collaborates with the elongation factor G, and help to destabilize that ribosome, releasing either the 50S first or the entire, disassembling the entire ribosomal subunit as part of it. So then you have, this is kind of summarizing what I just said, right? You have the termination of translation, there's a stop codon, one of the release factors positioned here with the help of GTP and other types of protein. This becomes destabilized, and it's really kind of forces a change in conformation that's really happening biochemically by adding all these additional protein complexes, and just destabilizes this complex. A peptidyl tRNA cleavage will happen using, and there's going to be a lot of enzymes that's part of this process that will literally cleave the polypeptide off of the tRNA that's in this P site and release that polypeptide that you have, disassemble the ribosomes, tRNA gets recycled, the mRNA is now free, and you can terminate translation that way. So in terms of when you think about all of so that's where I want to end in terms of translation termination and those factors that are important, is to really think about the difference between the translation machinery in eukaryotes versus prokaryotes, right? Because you will be doing a lot of these types of comparisons. So what are the components of the ribosomal subunits, tRNAs, initiation factors, what facilitates the start of translation in eukaryotes versus prokaryotes? I mentioned, right, understanding, and we talked about this the last time, what are two events that must occur prior to protein for the protein to even be synthesized, and having an idea what the Shine-Delgarno sequence and CoDES sequence are, learning to compare and contrast their roles, right? And that's really, they're working in two different organisms and what they do in those two different organisms. What makes up that initiation complex, right? So you need that initial initiation complex to be established. And elongation and termination, the two processes that are very similar between eukaryotes and prokaryotes, right? It's just different proteins that are involved in different names, but the idea is the same. I want you guys to also understand the genetic code, right? Knowing what a start codon is, a stop codon, what are frame shift mutations. And in the context of genetic code, as I mentioned the last time, understanding how to read how to read an mRNA sequence and determine what the codons will be. Determine what the codons will be if the code was non-overlapping or if the code was overlapping, right? So imagine having to do an exercise like that. And also being able to pull together all the different levels of gene regulation. So knowing that, okay, here is your DNA sequence. How do you transcribe that DNA sequence to RNA? And then that RNA to a protein. And you will be given the charts, so you don't need to memorize the codons, as I mentioned, or the amino acids for those codons. And understanding orientation, right? Which strand is being transcribed and how is that being translated? What is the direction of the protein that's being synthesized? So really trying to focus in on pulling all those three different levels together, okay? So this is really, again, another summary talking about the difference between translation and initiation, right? Understanding these two complex formation, which I think will really help you. And if you start here, it will really help you to understand these steps and see how simplified it is, right? I'm not going to ask you on, what happens when an ATP comes in and what's left and these type of things. It's really more larger concepts. You need your 40S. How is the ribosome recognizing the mRNA? What unique features in the mRNA allows the ribosome to recognize it? And also the different initiation factors. Some of them have very specific roles. So really understanding what the roles are for the initiation factors would be helpful. And both of these complexes form. What's interesting is that once you start translation of an mRNA, usually you have multiple ribosomes that's attached to that mRNA. Okay, so it's not just one ribosome comes in and then translate that mRNAs. Quite often, mRNA molecules have multiple ribosomes attached to it, and they're seen as polyribosomes or polysomes. And this is an electron micrograph showing actually in a cell what this structure looks like. So you have your ribosomes that you can see here, that looks like these little bubbles, and it's attached to a mRNA that's being translated. And really, as you can imagine 5' to 3' of the RNA, so you could imagine that this was the first ribosome that gets attached. Once you have enough peptides being made or amino acids being added, and this ribosome starts to move along, another one comes in, right? So you're going, you're adding this way, so this is the very first one, okay? And it just keeps adding and translating this messenger RNA, so it's kind of in a polyribosome format. The other thing that is really important here is that for most messenger RNAs or most mRNAs, it takes about 20 seconds to several minutes for you to be able to synthesize most messenger RNAs. Because you're able to have so many of the ribosomes attached to the mRNA, you can really control the speed as well of overall rate of protein synthesis, right? Depending on again, it really depends on the mRNA and particularly what that gene is that you're trying to make that protein for, okay? So I told you, you have your protein made now, right? So you think, okay, you went through all of these steps, you replicated your DNA perfectly, you transcribed your coding sequence perfectly, you've processed your messenger RNA, you processed it perfectly, it got out into the cytoplasm, it was able to be translated perfectly. That's not really the end, right? So once that protein is released from the ribosomes, most proteins are unable to function, okay? In order for them to become functional, they need to be folded properly. And this folding of the proteins to make it functional is called a post-translational modification event, okay, because it's after translation has occurred. And the most important of these post-translational events, of course, is the folding of the newly synthesized protein. And it needs to be folded into its correct structure or conformation to be able to function in the sense that a lot of proteins interact with either DNA directly or they interact with other proteins, right? So they need to be folded properly to be able to facilitate those interactions that's needed. A lot of proteins also need to go to certain places within the cell. So they also need to be able to fold properly to get there. So a protein that is folded correctly is considered to be in its native form, as you can see here. And an unfolded or misfolded protein is said to be in its non-native form. In order for this protein to fold, it can fold either through non-covalent bond formation, right? So imagine that you have your polypeptide chain, right? You can have folding with certain types of cofactors that will bind to that protein or to that amino acid in that protein and help to fold that protein into the conformation it needs to be in. And this binding of the cofactor helps to assemble the different parts of that protein so that it can interact with different parts of that polypeptide chain. You can also have covalent modifications, right, of certain amino acid, and this is what you see with this blue and yellow, right? So you can either have a phosphorylation that occurs, and we talked about phosphorylation when we were talking about chromatin level modifications, right? Where that was also a post-translational modification where you're adding a phosphorate group to a particular amino acid on those histone tails, those N-terminal histone tails, right? So phosphorylation is it's a very important modification for protein function. And that can be added. You can also have glycosylation where you're adding a glycosyl group to the protein as well, which is shown here in blue. And the addition of these will also recruit other types of protein to make this particular complex. There's more than 200 different types of covalent modifications, which we're not going to talk about all of them. The one that I think is really, really important is phosphorylation, and because this really helps a lot of protein to either become stabilized or can also alter the protein function in a negative way as well. Some proteins tend to fold while they're being synthesized, right? So it's still considered a post-translational modification because you need to have at least some of it already translated for this to happen, right? So some proteins will start to fold. So this red part is the polypeptide chain. You have your ribosomes attached to the mRNA. As it's coming out, this N-terminal part of the protein will start to fold immediately, okay? And then as the rest of it comes out, it will fold. So by the time the protein is released from the ribosome, it's completely folded and it can go on to do its function, okay? The way that these protein gets folded is with the help of other proteins, which we refer to as molecular chaperones. And these are proteins that really attach or bind to the protein to make sure that it changes its conformation into the right shape. It also helps to remove certain surfaces that might be exposed to the protein that causes a problem, and kind of shield those surfaces. So if you have really highly hydrophobic surfaces, sometimes that's not desirable, so you want to be able to shield that. So you kind of fold the protein to kind of protect that in some way. or in other cases, you need to expose those hydrophobic surfaces. So you need to fold the protein so that those surfaces becomes available, okay? So molecular chaperones will recognize these weird conformations or configuration. If in a case where you have hydrophobic surfaces exposed, which is normally not present because the protein is folded and it's hidden. The problem with some of these hydrophobic surfaces is that if they're exposed, they can form what we call aggregates. So it can aggregate with other proteins. And if you have an aggregation, that's really a problem because you can't really use that protein for anything, right? It's an aggregate, it's no longer functional, it cannot get to do what it needs to do. Protein aggregates can, in some cases, particularly in humans, cause a lot of diseases that, I mean, a lot of inherited diseases are due to improper folding of protein and these aggregate formation. And they can really cause severe symptoms and even lead to death in some cases, depending on the type, depending on the protein that gets aggregated. So molecular chaperones are really, really important in the cell for helping to make sure that these proteins get folded the way that they're supposed to. And in the two that we're going to really talk about here is these heat shock type of proteins, and they're very well studied chaperone proteins. They're we've seen them in bacteria when we were talking about the way in which you can regulate translation efficiency, right? So in bacteria, we talked about the fact that when you change the temperature in bacteria, you need to trigger the production of these heat shock proteins that are usually only activated under this heat stress, right? And they're activated on this heat stress because the sigma factor that makes these proteins is usually only active under heat stress. So in terms of the way that these heat shock proteins can work as molecular chaperones, there are two major families that we often study, and within these families there's multiple proteins, but there's HSP60 and HSP70 proteins. They function very similarly. There are some differences, as you'll see, but they each work with another set of associated proteins to really help with the folding. So they are able to bind to the exposed hydrophobic patches on an incompletely folded protein, use the help of ATP, right, hydrolyzed ATP, and often binding and releasing the protein substrate with each cycle of ATP hydrolysis. Okay, so it really helps to fold and then release the protein in this proper, supposedly proper conformation. You also have, depending on the organelles, whether we're looking at mitochondria, for instance, you have different types of these heat shock proteins that function as chaperones in that context as well. So HSP70 is a chaperone that works really early in the life of many proteins, often before the protein leaves the ribosome. So remember I told you can have this folding that's occurring as the protein is leaving the ribosome, right? So this is something that HSP70 is able to do. So as the protein is coming out of the ribosome with the help of ATP, this protein is able to bind to this this chaperone is able to bind to this protein and help start the folding, right? So it really binds and by recognizing those hydrophobic surfaces and really help to bind and shape the protein into the right conformation. Okay, and every time you have ATP hydrolysis that happens, because ATP, it's kind of, it will hydrolyze very rapidly and very quickly and randomly. So once that happens, the protein gets released if it's correct and when it's correctly folded. This process can also happen, can result in incorrectly folded proteins because either you don't have enough time or for some reason something went wrong and it's not folded properly. they will go back into this cycle again, right? HSP 70 is around. It will take up this incorrectly folded protein and try to fold it again, okay? HSP 60, on the other hand, works more with protein that has been fully synthesized, so it's been released from the ribosome. And the way that this protein works is because of the way that it's shaped, right? So it have this kind of barrel shape where you have these structures and you kind of have a lid. And it's often referred to as a chaperoning because you can have, it's a chaperone, you shuttle your protein into one part of this HSP 60, and most often it's just one part that's involved in the folding, so the top half and not necessarily the bottom half. And once it goes into this this barrel, with the help of ATP, a lid is really put onto this chamber. Again, these are all molecular protein context, right? So it's not really a lid, but it's kind of a cap, something that seals the structure of this HSP 60. So the protein is in there. The protein will be folded, right, within this chamber. And when ATP hydrolyzes, the lid pops off from that force and energy of ATP hydrolysis, and then that protein gets released, okay? So this is, again, this is a process that occurs once you have a fully synthesized protein for this to happen. And these are really just the steps that I just mentioned here instead of repeating everything. So the steps are here to describe this entire process. So even though a protein can fold and you have all of these checks to make sure that the protein gets folded either at a very beginning stages or once everything is already synthesized, you can still, this is not a completely foolproof process. So you can have some proteins that are not able to fold, okay? And even after you refold it many times, as you can see with HSP 70, where it was trying to refold the protein that wasn't folded properly, they still are not able to fold. And they, in the end, either form these protein aggregates, or one of the major pathways for proteins that are incompletely folded after multiple rounds of trying to fold them properly is for them to be degraded or digested, okay? And they get destroyed by a protein, and because it's a protein, it's a proteolysis reaction, and the mechanism or pathway that's involved in destroying these incompletely folded or improperly folded protein is referred to as the proteasome, okay? And this proteasome is an enzyme that really just cleaves the proteins and chews up and destroys it, okay? It is a structure that's kind of made up of this cylinder, that's shown here in yellow, with 4 rings around it. And then on each side of this cylinder, you also have parts that was referred to as a cap, okay? And the 20S and 19S, again, is just the sedimentation rate of these protein complexes. And you have these attachments on either side of this ring that helps to either seal the protein in or attach the protein, puts it through this chamber and then releases it out. And they're all involved in that process of digesting that protein. So this is kind of a schematic describing how that actually works, right? So this is really just the proteasome we're looking at, right? So you have, as I mentioned, you have this central cylinder, which is the protease, right? You have the active site for the enzyme for the protease. And then you have these 2 caps at the top and the bottom. And you have a protein that comes in. in order for the proteasome to recognize a protein that has not been folded properly, there needs to be something on the protein, right? It needs to know that this protein has failed the folding step, right? Because otherwise it will degrade protein that were just released and just haven't had time to be folded yet. So in order for the proteasome to know that this protein is a protein that needs to be destroyed because we cannot fold it, there's usually markers that are present on that protein, and the markers are these polyubiquitin chains that's shown here in red. And they get attached to these proteins that are not folded properly, and you'll see a little bit of that in a minute. So they get attached to these proteins. The proteasome recognizes this polyubiquitin chain because it has the receptors. It's present in this cap, pulls it into this chamber, cleaves off the polypeptide, right? So the ubiquitin gets cleaved off from the protein, gets recycled to go and attach to another unfolded protein. And the protein gets pulled into this chamber, and the protease just chews it up. And you have this, what you see by these little dots, and it gets released and destroy the protein. Okay. So it's really just sensing these proteins that needs to be digested because it have this polyubiquitin markers on there. And it's able to do that simply because of the way that the structure of this cap is, right? This structure has ubiquitin receptors. It recognizes these polyubiquitin chains that's there. And it also have the enzymes that are important for cleaving this polyubiquitin off of this protein before it goes into this main chamber. Okay. So the protein is not folded properly. With many attempts to get it folded, it gets destroyed by this proteasome, which is this multi-protein complex that really destroys these unfolded proteins. In the cell, sometimes you need to be able to, you want to turn over protein very quickly, okay? So this type of destruction is referred to as regulated protein destruction, okay? So these are proteins that you only want to be produced at a certain time, and then you don't want it to be produced anymore, okay? If there's mRNAs that are around, then it will keep being translated and making that protein until you've used up the mRNAs or you've degraded the mRNAs to turn it off, okay? So those type of proteins are highly regulated. And if you can imagine the type of proteins, like for example, proteins that are involved in mitosis, right? They just need to be present during mitosis, but as soon as the cell goes through mitosis, you need to get rid of those proteins, right? And that's kind of the way the cell does that, right? Is by doing this regulated protein destruction. So short-lived proteins are degraded rapidly at all times, right? So they're just keep getting degraded. Then you have conditionally short-lived proteins that are usually stable under some conditions, but then depending on what happens in the cell, as change in the condition of the cell, these proteins become unstable and will then get destroyed, okay? So that's a way that you can regulate the protein that's around in the cell. The way that the cell knows that these proteins need to be destroyed because you don't need it anymore, right, to really regulate them, is that you also have to have markers on these proteins in order for that to happen. We talked about the fact that the proteins that are not folded properly gets degraded by the proteasome because they have this polyubiquitin chain that's present on the protein, which the proteasome recognizes, right? So in regulated destruction of proteins, there's usually two general ways that this can happen, and is where you actually add a ubiquitin molecule to the protein itself. And that involves the use of these E3 ligases, right? So it's an E3 ubiquitin ligase, and it's a ligase, so it adds something, right? It's ligating something onto a molecule. So you can have a case where you have this E3 ligase, it gets phosphorylated by a protein kinase, and this phosphorylation by this protein kinase is enough to trigger it to ubiquinate a particular protein. You can also have a molecule binding to this ubiquitin ligase and changing the conformation. So it's an allosteric transition, right? Something binds to it and makes it active to bind to a particular molecule. Or you can have another type, not necessarily a ligand, but you can also have it interacting with another type of protein, a larger protein, for example, that then triggers this transition into an active form, okay? And then it's able to attach to a molecule and activate it, okay? So this is one way, is by activating using these ubiquitin ligase so that it can add a ubiquitin group to your protein. The other way is you can create or you can uncover or unmask a signal that might normally be present on these protein, but it's hidden, right? And when the cell needs to turn it over or degrade it, these signals need to be unleashed for the cell to recognize, okay, this needs to be degraded, okay? So by doing this, you're kind of unmasking a signal that's usually otherwise hidden. And once this signal is revealed, it's able to bind to this ubiquitin ligase and adds a polyubiquitin chain to it, and it gets degraded, okay? So in this case, you're activating a degradation signal, right? Comparing to activating of a ubiquitin ligase to add it, okay? So you have your protein, there might have been a signal due to, some kind of kinase coming in. This kinase can phosphorylate a particular site. When it phosphorylates this particular site by this protein kinase, it's able to recruit a ubiquitin ligase to this site. The other way is within the protein of itself, you will have an attachment of a molecule to this protein, maybe through interaction with this other molecule, where this site that recruits the ubiquitin ligase is hidden. You can cleave or dissociate, more likely dissociate this mask or this molecule from this region of the protein. And you can dissociate it by changing the composition within the cell. Okay, so maybe certain level of maybe just changing a pH can cause this protein to dissociate from this region and unmask it. You can also cleave off in a case where you have a protein, you have that signal that needs to be ubiquitinated, but it's protected because you have a protein directly attached to it. You can cleave this protein off using an enzyme and then expose this region that gets polyubiquinated. Okay, so either through activation of the ubiquitin ligase that will then add the ubiquitin group to your protein, or you unmask a signal that's already in the protein to then help to recruit these ubiquitin ligase to add the polypeptide chain, that polyubiquitin chain to it. Okay. So the why in these cases where you have phosphorylation, in both of these cases, phosphorylation by protein kinases that help in this process. We know that phosphorylation, as I mentioned, is really critical in changing the stability of proteins. Kinases are the ones that add these phosphate groups to those proteins, and phosphatases are what remove these phosphate groups, right? So it's something that's reversible. And you can have, if you add the phosphate group to some amino acid, this will help to recruit particular ligases to that site and help to polyubiquitinize that particular protein that has been phosphorylated and help to destroy it. So really, a lot of the post-translational modifications that we understand when we study protein and translation and post-translational modifications is really based on what we understand from phosphorylation. I mentioned there's more than 200 different types of modification, but phosphorylation is one that's been really well studied in the cell because we know the players, the enzymes, we know how they work. And we also know the amino acids that are more prone to be phosphorylated, right? When we talked about chromatin level modification, we were talking about phosphorylation of certain, serines. We saw that when we were talking about transcription, initiation to elongation, right? That C-terminal domain of that polymerase needed to be phosphorylated in order for you to really trigger that elongation. The amino acid on that C-terminal domain that was being phosphorylated were serines, okay? So there's particular amino acids that are highly susceptible to being phosphorylated. So not all amino acid is really happening too. Okay, but serine and threonine are kind of the ones that are really most highly phosphorylated. So that is that step, right? So you say, okay, now you troubleshoot all of your different steps in regulation, right? So you went through this protein quality check, it's folded properly, it's functional. The ones that are not folded properly gets degraded. The ones that need to be turned over gets turned over. That is not the end of quality control mechanisms, right? To prevent, protein function. You can also control protein, you can also control translation by controlling damaged mRNAs. Okay, so we're taking a step back, right, from post-translational modifications and now looking at the mRNA that gets translated. So we went through a lot of different steps where you're processing mRNA. We talked about the fact that you need to add a 5' cap, you need to add a poly-A tail. You also need to make sure that your mRNAs are properly spliced, right? It's processed mRNA. When all of these steps happen, and then the mRNA, looks stable, right? Because those are the different modifications that prevent the stability. That helps to stabilize the mRNA and prevents the degradation. Sorry about that. You have your mRNA goes into the cytoplasm. It needs to be translated. The mRNA is not naked when it leaves the nucleus and goes into the cytoplasm. It's bound by these proteins, right? It's bound by the poly-A binding protein. It's bound by proteins that are 5' cap. It's bound by proteins that attach to the regions where you just spliced out an intron, right? So there's proteins that are bound to those 2 exon junctions, or at that particular exon junction, okay? So there's a lot of proteins that are attached to this mRNA. It's not naked as it makes its way out into the cytoplasm. However, you can have mRNAs that are not properly processed that makes it out into the cytoplasm to be translated, right? And once it's out into the cytoplasm, it's there, the cell assumes it's being processed, and it needs to be translated, okay? In addition to that, you also have mRNAs that, as they're leaving the nucleus, right, they're going through that nuclear pore to get to the cytoplasm, they can become broken or damaged, yeah, either through that process or when they arrive in the cytoplasm. In order for all of these steps to tell the cell, okay, this mRNA can at least start to be translated, I should say, there's proteins within, surveillance proteins within the cell, in the cytoplasm, that looks for certain features on the mRNA. It looks for the 5' cap, and it can recognize the 5' cap because it recognizes the protein that's bound to that 5' cap. It recognizes the poly A tail that's there because it recognizes the protein that's bound to the poly A tail. It recognizes that this mRNA has also been spliced because it has these proteins that says, okay, it's being properly spliced. And once you have this there, the surveillance mechanism will scan this mRNA. If that mRNA does not have all of the proteins or all of the features of a protein that has been properly processed, like one of these different steps, it gets degraded. And the way it gets degraded is by this process called nonsense mediated mRNA decay, okay? So it's reading the mRNA as kind of nonsense, right? So, and it's degrading it, okay? It's a process that involves multiple other proteins. We're not going to talk about what those proteins are, but there's many proteins that are involved in this process. So it will scan the mRNA looking for these signatures of a properly processed mRNA. If it's not, it will just degrade it, okay? One particular signature that this surveillance mechanism looks for is usually if your mRNA have a stop codon in the wrong place, okay? You remember when we were talking about frame shift mutations or translating a mRNA in a different frame than the normal frame? What can happen is that when you do that, as a result, if you're in a different frame, you could encounter a stop codon before that mRNA needs to be terminated, right? Before that normal stop codon would be, right? So this can happen, right? Exactly, where you have, as Sheila said, pre-termination, right? This can happen, right? The other way that you can have a problem with a stop codon being where it's not supposed to be is if you have incorrect splicing. So within introns, there's usually a lot of stop codons. I shouldn't say a lot of. You can find stop codons in introns. If for some reason that intron is not spliced out by accident, then you have a stop codon there now, okay? So the cell will notice that, okay, this is a stop codon that shouldn't be there, right? So that's another way that you can alter, the cell will recognize that these mRNAs are not great and they shouldn't be translated. So I'm going to just run through the steps and then I'm going to show you a graphic because it's just easier that way. So the first thing that happened is that this nonsense mediated mRNA decay mechanism, which is composed of a number of different proteins, usually begins as the mRNA molecule is being transported from the nucleus to the cytosol. Okay, so it's coming through that nuclear pore into the cytoplasm. Okay, so it starts already looking to see is everything there. when the 5' end emerges from that nuclear pore, the mRNA is met by the ribosome, right, to begin translation. Okay, so this needs to happen. So it needs to recognize that 5' cap is there in order for it to start translating. As translation proceeds, the ribosome recognizes these, what we call exon junction complexes. These exon junction complexes are proteins, multiple proteins that are present on the region between two exons that have come together because you've spliced out an intron. Okay, so the two exons will come together. And there's usually put the proteins there. And that's really a signal to say that this mRNA has been spliced. Okay, and again, this gets recognized. And if for some reason you have a normal stop codon which will lie within the last exon, right? So that the ribosome reaches it, it already scans along past all of these exon junction complex proteins. And it reaches it and stalls because this is the proper stop codon. This is where we're stalling. This is your terminating translation. Everything is fine. So this mRNA passes this inspection process and really gets released into the cytosol for it to be continuously translated. So when you have an mRNA that's coming through the nuclear pole into the cytosol and the ribosome is scanning it, what is happening is what we refer to as a first round of translation or a pioneer round of translation. This pioneer round of translation is what's telling you that this mRNA is good. There's a 5' cap, the exon junction complexes are present, we reach the right stop codon. This is not something that's really taught a lot in classes or we think about on a regular basis because we just think mRNA is made, it goes into the cytoplasm and it gets translated. Before that full protein gets translated and you keep making more of that protein, there is an initial round of translation, which is really inspecting that mRNA to make sure that it's okay. And if it's not okay, it gets degraded, okay? So when that initial first round of translation is happening, if for some reason the ribosome reaches a stop codon earlier, either due to weird alternative splicing or a frame shift mutation, the mRNA molecule will get rapidly degraded, okay, by this nonsense mediated decay pathway. Because it's stalled, the ribosome stalls, and this nonsense mediated decay recognizes this stalled ribosome before it reaches the end of the mRNA, and it degrades it. And it degrades it by adding exon nucleases or endonucleases to chew up the mRNA. And there's many of them in the cell. Some are specific to the 5' end, some are specific to the 3' end, but it really just degrades the mRNA this way. So that's the first round of translation that really allows the cell to test the fitness of each mRNA molecule as it exits the nucleus, and this is what's referred to as a pioneer round of translation. Okay. So this is a step that's really often hidden, and again, we don't discuss it a whole lot because it's a check for the cell to determine whether or not you need to degrade it. So you have, and this is a schematic, just kind of summarizing exactly what I just said, right? You have your mRNA. Start codon, I said you can have stop codons within introns, and then you have your normal stop codon at the end of the region that's coding within your mRNA. If this is spliced properly, right, normal splicing, your mRNA, there's proteins, these exon junction protein complexes that gets attached right at these regions, okay? You'll have proteins at the 5' cap, you'll have proteins at your poly A tail that's present. Okay. Abnormal splicing, this remains, and then you have this exon junction complex, downstream. If for some reason, as this is coming through the nuclear pore complex, you have this pioneer round of translation that's happening, the ribosome comes in, it first checks that it gets recruited there because you have the 5' cap, right? And eukaryotes, you need that 5' cap to help to recruit that ribosome, right? So it comes in, it scans along the mRNA, looks for the start codon, right? The preferable start codon because it has all the perfect Kozak rules, etc. And it scans along this mRNA, meets with these exon junction complex proteins, removes them because it's just a marker, again, it's been properly spliced, removes it and scans along, it encounters the last stop codon. And once it encounters this last stop codon, you terminate translation, right? So mRNA survives, efficient translation. There's a whole range of interactions that can also happen, that also happens, right? So for some proteins or for some mRNAs where the coding sequence is pretty short, you can imagine that you can ask yourself, how does the ribosome know that, okay, this is the real stop codon, right? Compared to something else. There is, to some extent, interactions that can happen with proteins that are somewhere in this poly A tail that also helps to facilitate this region to say, okay, this is towards the end, okay? And once this happens, again, the ribosome can recognize it, again, with the help of all these other signaling molecules, and this mRNA is past this inspection check and gets moved in, like, completely into the cytoplasm now, and you can translate it as many times as you want until you don't need it anymore, until you don't need that protein anymore, or you've used up all the mRNA, it's no longer being transcribed. In the case where you have a abnormal splicing, so you have the stop codon, the intron is still retained, there's a stop codon here. As the ribosome moves along this mRNA, it recognizes that there's a stop codon. It also has not passed any exon junction complexes. And also you have in the exon junction proteins that are downstream, there are proteins that are attached to it that also interacts with this ribosome to say, hey, this is a premature termination codon. There's actually another region that has been spliced out, and that the stop codon is downstream of that. Yeah, I'm saying this in a simplistic way, but it's really, as the ribosome stalls, all of this is happening at the same time. It stalls on this premature termination codon. The downstream exon junction proteins recruits additional proteins, which are called, in this case, upshift proteins, but there's also other proteins that are part of this process, and interacts with the ribosome to say, hey, this is something is weird here, right? This is not supposed to be, this mRNA is not supposed to be translated because this wrong splice, the splice, it's not spliced correctly, okay? And then this really helps to trigger this nonsense mediated decay, because these proteins will then recruit the enzymes needed to really chew up that messenger RNA. Okay, so another quality check mechanism, depending on if there's a stop codon, to make sure you're not making the wrong protein. So after you've gone through all of these quality checks to make sure that your protein is great, it's the right protein, there's no truncation, it's being properly folded. How does the protein know where to go? I mean, all of these proteins are being produced in the cytoplasm, but some proteins, I mean, obviously transcription factors, right? They work in the nucleus. Some proteins work in particular membranes. Some proteins work in particular organelles and compartments, right? Some proteins work in the ER, right? How do the proteins know where to go? Okay. So the final level, like the amount of protein that gets produced in a cell, really depends upon the efficiency of each step, right? And you can see that, right? You can see that if you mess up transcription initiation, right? And this was the step where we talked about how you can use nuclear RON assays to look at transcription initiation and the efficiency of transcription, right? And if you mess up the initiation of transcription, you will have less transcripts and as a result, less protein, right? If you mess up the processing of your message, right? We're not going to talk a lot about degradation of mRNA, right? You can also have degradation of mRNAs that are happening even before this step, right? Of the pre-mRNA, which gets degraded. But you can imagine that at this step as well, capping, elongation, splicing. If something goes wrong, you also affect the final outcome of the protein. If you mess up the way that that protein gets to the cytoplasm, right, through the export process, right, you can also affect the final amount of your protein, the level of your protein. And once it gets out into the cytoplasm, during that initial first round of translation, that pioneer round, it can also be regulated at this point, okay? Your protein is made, it passed this step, folding, right? You can, if something goes wrong with the folding, you can also degrade your protein, right? So in the end, your final protein level had to have gone through and survived all of these different steps of processing, right, to be functional, right? So it's not an easy process in the cell, and that's why it costs so much energy to do this. That cells are really selective on what needs to be transcribed or what needs to be translated, right? Because it's really an intensive process that's happening, okay? So that final level of each protein is really the balance between the rates of synthesis and degradation all throughout these steps that will determine what that final protein abundance or level is. So proteins know once they get to this step and you have your protein properly folded and it survived everything, now they need to know where to go to actually do their job, right? So within proteins, already embedded in the DNA sequence are certain sequences that codes for signals within the protein, okay? So when the protein gets made, it said they're kind of synthesized with these sequences that kind of what we refer to as barcodes, right? It tells the protein where to go within the cell, okay? So you can imagine here you have your mRNA, you have your protein, your ribosome making your protein. That protein gets made, yeah? And once your protein gets made, part of this protein is some kind of a signal. In this case, this is a signal peptidase, right? And this signal peptidase is a protein that needs to be attached to the plasma membrane, right? So it needs to be attached to this plasma membrane. And it is synthesized as part of this protein or this polypeptide. Once it's synthesized and it goes where it needs to go, this signal peptide will be cleaved and released, and the protein then goes to where it needs to be. And in this case, it needs to be attached to the membrane, right? Proteins that for example, proteins that need to function in the nucleus, such as RNA, DNA polymerases, right? Because transcription is happening in the nucleus, replication is happening in the nucleus. Transcription factors, which helps to regulate transcription. These proteins, they're made in the cytoplasm, but they need to get back into the nucleus to do whatever they need to do, right? Because that's where all of your DNA is, right? And in order for this to happen, these proteins have a signal that's referred to as a nuclear localization sequence, okay, or an NLS. These proteins have this signal that allows it to go back into the nucleus after it's been made in the cytoplasm, okay? It also helps, there's also signals on these proteins that tells the protein you need to get out of the nucleus into the cytoplasm, okay, after the fact. So there's also nuclear export sequences on some of these proteins, okay? If you have a protein that you want to study, and that protein, you check the sequence because we know what these sequences look like. I mean, there's enough research, there's enough work done on these different protein sequences, so we know what they are, right? If you have a protein, or let's say you have a gene that's going to be made, that's going to make a particular protein, and you want to study how this gene might function in the nucleus, because it doesn't have a nuclear localization signal, this gene might function. Let's say this gene functions in the cytoplasm. That's where it's supposed to function, right? You can attach a nuclear localization signal by cloning, right? So you can clone your gene using either PCR or some other type of method, attach a nuclear localization signal because you know what that is. The nuclear localization signal is just a sequence, right? It's just a sequence. It's really a DNA sequence that you will attach, and that will be transcribed and then gets translated, okay? And that becomes part of your protein, and it will take it back into the nucleus. And we quite often do this. We quite often modify the nuclear localization signal to see what happens when a protein is no longer able to function within the nucleus. We can do that because we can also attach a fluorescent protein so we can track within the cell where that protein is at any given time, okay? So that's really the final outcome of a protein once it's made, okay? So you're going from your DNA to this protein, it needs to be targeted where it needs to work. And that's really the final step of this level of gene regulation that we're going to talk about, yeah? So then next class, we'll talk about how do you look at, and we'll also do compare and contrast. How do you look at how much protein is there, or how do you study the amount of protein that's present within the cell compared to the amount of mRNA and these type of things. So we will discuss those methods on Thursday. For this part, what I want you guys to get out of it, again, understanding that protein needs to be folded to function properly. The two molecular chaperones, HSP 60 and 70. Again, the whole details about how they work are not super important. What's important is when they actually function, right? Because they'll function at different times. And what happens when you cannot fold the protein properly? What happens to that protein? And also, how do you also regulate the production of functional protein at the mRNA level, okay, during that first round of translation? So understanding what's happening at that pioneer round of translation is important. And then again, very basic understanding that proteins will have embedded within them the signals for it to target where it needs to work or where the function needs to go. Okay. And then this is pretty much the last slide again, questions that I want you to think about, you know, what is post-translational processing and why is it important for protein function, right? And if I was to ask you this question, you would definitely tell me, oh, you know, some proteins cannot function unless it's being folded properly or modified in some way. And in the case of, you can add a phosphate group, right? Phosphate group will help to fold properly so that it can be properly transported where it needs to go. Or I mean, you also want to be able to destroy proteins that don't need to, that shouldn't be around in the cell anymore. You also need to regulate protein turnover, right? So proteins that you don't want to be around, or they're good proteins, but you just don't want them to be around anymore, right? So you want to regulate their production, and you regulate their production by just destroying them once you don't need them anymore. And you can destroy them in either of two ways, right? By either activating the E3 ligases to add this polyubiquitin that's recognized by the proteasome, or by unmasking a region within that protein that then gets phosphorylated, which attracts the E3 ligase complex as well, and helps to add that to it as well. So we have 4 minutes. We can talk about anything if you guys have any questions.
For that question, Professor, where it says what is post-translational processing, would like an acceptable answer just be like modifications done to the protein after it has been translated from mRNA that allows it to continue towards its protein targeting, like something along those lines.
That would be a very acceptable answer, yes.
Okay. And then I had another question about the heat shock protein 70. Let me see. Heat shock protein 16 is when the protein's already detached from the ribosome, but heat shock protein 70, is that when it's still?
Right, so 70, so if you were to compare and contrast these two proteins, you would think HSP 70 acts early in the life of many proteins. So it starts folding the protein as it's being synthesized, compared to HSP 60, which functions later, right? So you have to already make all of the protein, it's already detached from the ribosome, and then you fold it. So HSP 60 functions later, and HSP 70 functions earlier. Yeah. That's clear?
Yes, thank you.
Any other questions?
Yeah, I do have a question about the nonsense.
Let me stop the recording. Someone just reminded me, so we don't have this issue. 1 second. Stop recording.